But now I'd like to introduce Dr. Gerard, who is a clinical senior lecturer and honorary consultant at Queen's University in Belfast. He's also just flown in from Puerto Rico, I think you told me. Yes, so be gentle with him, because this chap's got like jet lag. But he's also going to talk to us now about clinical trials and make it very interesting. No.
No pressure there at all. Yes. So I don't know what time zone I'm in at the moment, so forgive me if I make any spoonerisms. I've been told 45 minutes still, Deborah. So I've started my stopwatch and I'm aiming for 45 minutes. My slides are not on the screen. I can do this talk again. It was really good. It won't be as good the second time. Okay, I see someone's moving down here. Yep, great. Thanks, folks. All right. Crisis averted. Slides are coming. Yeah, so I'll introduce in the meantime. Effectively, what I want to do is bust some jargon. I think there's a lot of literature out there and scientific research which would be interesting to be able to understand and hopefully informative, but it's not really written in a way that most people understand, including some clinicians. And you know, as doctors, we spend lots of time trying to learn about how to understand clinical trials and research in general, and also the process of research, because it's not like anything else that goes on in medicine. So for the BTOG conference this year in Belfast, where I practice, we designed this talk, and I think the feedback was quite good. So we made some amendments, and we thought it would be useful to run here as well. So hopefully about 30 minutes or so on the slides and content, and then after that we'll have time for questions. So planning to cover how does clinical research that happens in people get to the patient? How does it get to the patient? How do we do the research? It's number 2. And number 3, how do we understand how the research is written down? How is it communicated? So first of all, how does clinical research get to the public, the patients, families? Obviously, the research is to do with the patient. The patient is the most important part of the research, but the patient is at the very end of a very long journey. And the journey for the research starts with usually clinical trials. And clinical trials are simply broken down in probably most commonly as phase 1, phase 2, and phase 3 trials. And we'll speak a little bit about the differences in those in a minute or two. Often before the clinical trials have been organized, some little furry fellas have also taken part in some research as well. And maybe before that, some cells in a dish in the laboratory, and often even more superficial research before that, and looking at gene lists and other publicly available information. So that's the sort of technical part of research, and we will go into that in a little minute. But whenever research is going well and a new discovery is made, how does it get to the patient? Well, whenever a new result becomes available, often there's a press release, and this will be given out by the company or the organization who sponsored the research. And that sometimes even makes the news, even though these press releases have very little detail in them about the actual research. Then at a medical conference, often an international conference such as ASCO in the US, which is a global conference for clinicians and people who work in oncology throughout the world. Usually there, the lead author, the lead clinician who's been really involved in the research from the beginning, they will give a presentation on the actual nitty-gritty, the numbers, the statistics, the types of patients that are particularly applicable to this research. From that point onwards, there's usually a lot of excitement in the room. Often there's rounds of applause from clinicians and pharmacists and nurses who are attending the meeting, because it's like Christmas for us. We go to these meetings once or twice a year, and we're always hoping for a big change, a new treatment for our patients that we see. And sometimes whenever those moments are announced, it really is quite special. But the devil is always in the detail. And before we change practice, we need to see what the actual details of the trial are. And whenever it gets published as a formal paper, a manuscript in a medical journal, then we have the opportunity to run through all the details and see all the nuances of that study to work out how well was that study done. Obviously, anybody who's going to try and do some research to do with patients tries to do a good job to make sure the research is of a high quality. But sometimes, because there is so much going on in research, it's easy for the ball to be dropped on a few different matters. And so as a clinician trying to appraise that research to say, yes, you know what, I really believe that research, I take it at face value, you need to see the paper published. Once the paper is published, then lots of smart folk at a European or US level, like the FDA, they will get together in a room and discuss the paper and discuss the sort of advantages of the study, the benefits, the strong aspects of it, but also the weaknesses of the study. And then they'll also factor in the economical factors too, to an extent. Once there's approval at a European or US level, then here in the UK we have the NICE organization, which sort of runs a health technology appraisal process. So what does that mean? Well, effectively NICE are part of the government. They will look at the evidence in the study and the paper, and they'll say, yes, we believe that, or we don't believe that. They'll say, we believe it, but how much does it cost? And they have different ways of assessing that. And then if they're happy, it gets approved, and it becomes available in the NHS for doctors to prescribe. And that's how the new research eventually gets to the patient. How long do you think that process takes? Shout out a few numbers. 10 years, exactly. That's on average what you're talking. At least, which is quite shocking. So, number 2, how is research done in practice? So there's a lot of words on this slide, but I want to give you the detail because I do think it is important. There's 3 sort of phases to setting up a clinical study, a study done in patients. We have to first of all start with a hypothesis, an idea, a question, and that can be because you've done some research on animals, or you've noticed something on someone else's research, or you've done some genetic research from data from a different study somewhere else. But whatever sort of source your idea comes from, you've got to first of all think, well, is this relevant to the patient? And then we gather what data we can, and we build a team. We need people with the right skill sets. Is there radiotherapy involved? Is there genetics involved? Is there pathology involved? We find an organization to help us organize the study, and that's usually called a sponsor, and that can be a drug company, or it can be a statistics unit in a university, or even a hospital trust as well. And then we've got to think about the research question we're trying to answer. So we want to know, does drug X improve outcomes compared to drug Y? Well, we've got to think then, how are we going to design this study to answer that question? Once we've thought about the study design, then we'll write a protocol, so this will be the plan. And this plan is rigidly adhered to. We don't deviate from that. We can't halfway through change our minds and think, I don't like this result, I might change the population and bring it into this trial. You've got to stick to the plan. Then, once you've got a plan, money. And this is like everything else. Money makes the world go round. It's part of the research process as well. There's lots of people in lots of places who want to run trials. Some ideas are better than others. And effectively, because of the stringent budget, funders have got to decide which trial is going to answer the best question. And so we've got to make a really strong effort to put in a funding application that will be hopefully able to justify the funder giving my team the research money. And then once we have the funding, we can get some other approvals to do with ethics and governance and get the show on the road. And again, how long do you think that takes? Maybe 3-4 years, depending on how complex all that is. But again, that's after having done the animal research, some preliminary research, and that's before the 5 years the study takes, and then the 5 years after that to get the actual treatment to the patient, 2 or 3 years at least. So once we have the trial approved, we want to open that study and bring it to you, the patient, the family members, the public. there's a process for opening that trial at a hospital. It's not a case of protocol is received in someone's inbox and we say, yep, let's go. There's all sorts of hoops to jump through, and anybody who's involved in sort of professional working and managerial type work will be aware of how much paperwork goes into these things sometimes. It's all there for the right reasons, of course, but there's just so much red tape sometimes. Anyway, we're all driven to get this trial open at our hospital. We like the opportunity that this is going to open to some patients. We don't know if the treatment's better or not, but we've got a feeling and we want our patients to be part of that process. So we look for patients in our clinics. We have criteria, eligibility criteria, that we check each patient against. And if you are meeting those criteria, we can then approach you to talk about taking part in the trial. If the trial goes ahead for that person, then we have to document everything, absolutely everything. What color of socks you're wearing, not quite that detail, but everything else. And that's about side effects, trying to find side effects, trying to find problems, because we want that paper at the end product to be as detailed as possible and as accurate as possible. It would be unacceptable for a study to be supporting a new drug or a new treatment and then actually for new things to be discovered about that treatment after the treatment was given out in the NHS. That would not be right at all. During the trial, we'll also have problems. So sometimes unforeseen things happen and we've got to try and answer queries about trials inside each hospital and at a national level. And also during the follow-up period of the trial, we'll potentially ask people to give us samples. And that could be blood, could be tumor biopsies, it could be scans. And what those extra samples are for is trying to then, at a later point, once the trial is more or less finished, we'll take all those samples and analyze those and try and understand the biology of why treatment X was better than Y, or not better as well. And often it's from those samples that we then are inspired to answer the next trial. And it's through samples like that that we can understand, well, why does this drug not work for all patients? Oh, well, actually, it only helps people with mutation X. And then that branches off a new area of research about that type of mutation. And that's exactly what happened with EGFR back in the noughties. So once the study is done, all those pages and pages of information about your socks and everything else, they're all written up, they're all submitted nationally, the data is cleaned, and we have an analysis plan in terms of how to go through that data and report it. And again, the way we had a protocol for how to collect, how to find patients and give the treatment, and a protocol for collecting data, we also have a protocol for analyzing the data with statistics. And those statistics are also well suited to the type of research question that we're trying to answer. And then once we've got all that data collected and analyzed, that then starts off the press release, the conference presentation, the paper, and then those translational studies that I mentioned where we're looking at the samples. So that's the process at large. And more or less, the sort of common bits of research that you hear about most are phase 1, phase 2, and phase 3 studies. Phase 1 studies, really they're about collecting small numbers of patients together to try and assess the safety of a new treatment option. So those patients are often, they've used up all the options of treatments that are already available, and there's no other sort of tested options that are available. Sometimes those phase 1 studies are based on preclinical studies, that's the lab work, animals or cells in the laboratory. Once a phase 1 trial has confirmed the safety of a treatment, we then want some sort of feeling for, I've got a rogue timer here, we want some sort of sense for, could this treatment potentially be as good as the standard treatment? And that's where a phase 2 trial comes in. So a few more patients, not quite 10 or 20, but more like 20, 30, 40, 50, or even up to 100. And you're looking for a provisional signal that this treatment might actually be better than what we have so far. And if that's encouraging, then we move to a phase 3, 300, 500, 700 patients. And that's a head-to-head study of the new treatment compared to the previous treatment. Phase 3 trials are often randomized trials, so patients are randomly assigned to one treatment or another. And the idea of that is that you remove any sort of bias, any differences that might exist between the two patients and the two different treatments in the trial. because you don't want your result of the trial to be influenced by some other factor you haven't accounted for. So if you randomly assign patients to arm A or arm B, treatment 1, treatment 2, then you think that probably all the differences between the two arms, the two studies at the very end, that's just because of the treatment differences. Those are the common sorts of studies, phase 1, phase 2, and phase 3 trials. We also have these new emerging trial types, such as platform trials, basket trials, umbrella trials. This rogue timer is killing me. And then we also have observational studies as well, and biomarker studies too. So all of these studies are there, we can use them, but which one we use depends on the research question we're trying to answer. So for basket trials, for example, that's typically where you take a drug for, say, a mutation type, for example, ALK, and you bring in patients who have ALK mutations in many different types of cancer. Now, they're more or less only in lung cancer, so that's a bad example. But you could, for example, if there's another mutation that happens in different types of cancer, you would bring in different types of cancer to that one trial and give them all the same treatment. And then platform studies in the prostate world, there's a trial called Stampede, and that's a platform study where there's, I think, maybe about 15 different treatments now for different stages of prostate cancer, all organized within this one massive big study, this platform trial. And the reason we're trying to do that more and more now is because all that paperwork, the funding, the ethics, the approvals, that slow, tedious process that I mentioned earlier, well, platform trials effectively allow you to do that once for the study, and then you have lots of mini studies within that. So it accelerates the time to getting the research open and then getting the results out. So we're trying to use more novel approaches like that nowadays to try and get research expedited, to get it to the patients and the families a bit quicker. So, understanding research reports. I'm going to use an example study called the Pacific study in lung cancer. And what this study showed was that in people who get radiotherapy for localized cancer in the chest, often with chemotherapy at the same time, adding immunotherapy for a year afterwards improves outcomes. There may be some people in the room who've had this treatment in the past. Typically, we now don't use this treatment if an ALK rearrangement has been discovered, but that's been a more recent development. In this study called Pacific, which was the trial that sort of approved this addition of immunotherapy for a year after radiotherapy, patients with ALK rearrangement and EGFR mutations actually were included before we found out that actually this treatment might not be the best combination for those patients. So the Pacific trial was the first trial I ever seen presented at the World Lung Conference, which was in Toronto that year in 2017. I was a second-year registrar, was all excited about getting to go to Toronto for work reasons. Yes, I had a great time in Toronto. But the standout moment of that conference for me was whenever the results of the Pacific trial were presented for the first time. Because in lung cancer, stage 3 lung cancer, outcomes have been the same for years and years and years. And this was the first time ever that there was a 10% increase in survival overnight with this addition of this treatment. And the room erupted with laughter, tears, clapping. The clinicians were just so moved by the fact that we now have a better option for the patients with stage 3 disease. And it was instantly practice changing. So we're going to look a little bit into this paper just to sort of flag up some examples of how the data is presented and what it means and how you can understand it and apply it to other studies that you might read. To be clear, I don't think we all need to be reading studies all the time, but I think if someone recommends a paper to you to have a look at, it would be interesting and useful to be able to understand parts of it at least. So every paper that gets published has an abstract. And this is a bit like the blurb in the back of a book. It's just the headlines, the main story. Only this does give away the ending, unlike the blurb usually. And the abstract really is sort of just the need to know from the study which patients were in it, what was the treatment we did, what was the main result and what does it mean? That's usually on the front page, and a lot of the time I don't read a scientific paper when it comes out. I have a wee quick glance at the abstract, and sometimes that's enough to know what you need to know out of it. Sometimes it might help you decide, do I read this paper or do I not read this paper because it applies to me or it doesn't apply to me. Other things on the front page of the paper will be the actual journal name. So in this example, it was the 5-year data. It was in the Journal of Clinical Oncology. And you'll often see references written down if people are involved in helping form policies and things. You'll maybe see references for studies written like this at the bottom. And there's lots of numbers and details. A DOI is a digital object identifier. It's more or less a web link to the full paper. But you can also see the year and the journal name within the reference, the date that it came out, the volume and the issue, and then the page numbers too. It's like a way to reference this paper if you're ever using that while creating a patient leaflet or helping to, if a clinician comes to you and asks for your help to design a study or design a leaflet, this is the sort of thing you might see. Studies often nowadays come with like an interpretation box. This is a relatively new thing, so it's context for the study, again, like the abstract, but putting it into words. What is the point of this paper? Who cares? Who does it actually help? What's the main headline? And that's becoming more common now for studies. So often if the abstract looks very wordy or the text is tiny or you still can't really get a good feel for it from reading the abstract, reading the context box is a good way to sort of get a feel for what the study actually means. Often you'll also see on the front page information on the study registration. So all these studies, as I said, have to be carefully regulated. And there's a website called clinicaltrials.gov where lots of clinical trials are registered. So you can do searches there yourself. It's publicly available. People often go there to look for clinical trials that are ongoing or finished. You'll also see information about the sponsor, so who organized the study, and that can be important. This study was for a drug called durvalumab immunotherapy given for 1 year after radiotherapy. That drug is made by a company called AstraZeneca, and AstraZeneca organized the study. Now, obviously, as an optimist and, taking everybody at face value, I believe they've done a very good job, but a skeptic might think, isn't it interesting that this company found a really good, brilliant result for their new drug that's very expensive? So, we always have to sort of challenge these ideas, and I'm joking, of course, but, these things can be relevant, and that it's just worth looking at these details sometimes before you fully take a result at face value. The other thing that's also important is the people who wrote the report about the trial. So do they have any relationships with the company as well? So Professor Popat, who spoke last night, if he was an author in this study and he also receives loads of cash for doing talks for AstraZeneca, then you might raise an eyebrow when you see his name as the first author. So, those sort of things, again, are relevant. Again, that's not a rumor I'm trying to start here. Professor Papa doesn't do things like that. This isn't recorded, is it? No. So then moving on to the introduction, then the introduction is a sort of flowery part of the study, at the start of the study, which is setting the scene. And that's really just explaining what the problem is and where the evidence currently lies for treatments. The methods is where a lot of the nitty-gritty for a study can be found. In the methods section, you'll like to see things about the study design. Why did they pick the study design that they chose? You'll want to see what their main sort of end goal was, what was the sort of crux of the answer they were trying to formulate. That's called the end point of the study. And then you'll also see something about the statistical analysis too. And the statistics, the data comes out, there's, thousands of forms, as I mentioned, about data that's collected during the trial. But how it's all pulled together and interpreted depends on the statistics that are applied to that data. And therefore, that section of the paper is really important to read as well, especially if you've got an interest in statistics. So the study design for Pacific, just to run through it now, so patients who were performance status 0 or 1, who had a biopsy proving they had lung cancer, they were enrolled. They got radiotherapy, and that was usually 60 gray and 30 fractions, just an amount of radiotherapy. And if they didn't have any disease progression at the end of treatment, which is thankfully uncommon immediately, then they were able to get this treatment as long as any side effects from radiotherapy had resolved. And it specified any grade 2 side effects. So if patients had radiotherapy but still had a grade 1 reaction on their skin, for example, they could continue in the trial. And then patients were randomly assigned, so a random number generator in a computer somewhere decides whether folks are in one arm of the study or a different arm of the study. That's how patients were decided as to which treatment they received. And there was some background stratification for age and smoking history. And stratification just means that it wasn't a completely random process. There was some background to the computer program that made the allocation. And that's usually to sort of try and address clinically relevant factors. For example, taking smoking history into account would help try and tease out what the impact might be for people who have a mutation-driven cancer, since there's very light smoking histories or no smoking histories for patients like that. Older and younger patients is also an important factor. And then every method section usually talks about ethics and where ethics applied to and what sort of guidelines were followed. And that's really important too. In terms of the endpoints of the study, so the study wanted to look at overall survival, but also progression-free survival. And some patients would say, well, what's important to me is living the longest. Some people would say, what's important to me is not having relapse of my cancer for the longest. And there's no right or wrong answer, and that's something that we as clinicians struggle with. But we have to factor these things into our designs of our studies. And again, it's really important that that's stated up front so we can understand what the study is trying to tell us. What's also really important here is that they've specified that blinded independent central review was performed. And what BICR means, it means that the clinician giving the treatment didn't decide whether it was working or not. All the scans were sent to an independent person who doesn't know what treatment the patient was receiving, and they decide whether the treatment's working or not, which just makes it very neutral and objective in terms of reading out the response of that treatment. And that makes it fair, and there's no unconscious bias. It might be that if a clinician who's treating a patient and knows what they're getting has worked it out, even though the treatment's blinded, they might think, well, that scan, it's maybe a wee bit bigger, but no, I don't think it is. They're on that treatment. I think it's going to help them. And that might subconsciously influence what they record. So having blinded independent reviews is really good in a trial, and that's a strong sign of a good trial. We also see the authors usually write down what subgroups are important, and again, that influences how drugs get regulated and approved. And we also see some biological factors like PD-L1 status. And then lots of exploratory endpoints. So the top headline here is about overall survival and progression-free survival, but lots of other things like toxicity is also really important to clinicians, to patients and their families. So that again has to be declared in the methods. And then statistics, I don't think you need to understand the statistics at all. Most clinicians, to be honest, don't understand statistics very well after their exams. It sort of goes out the other ear once the exam's done. But we have to understand a basic level to sort of appraise the evidence. And we'll talk about a few of these things like time to event analyses and Kaplan-Meier curves. Intention to treat population is one important thing I want to flag. So in trials, we'll often enroll loads of people, but for some reason, 5–10% of patients don't get the treatment that we planned. And we don't know whether we should factor that into the analysis for trial or not. But the best thing to do is to include all patients who were planned to get the treatment, whether they got that treatment or not. That's just the fairest way to look at it. Because in reality, in the real world, when we want to offer a new treatment to a group of patients, again, some patients won't make it. So sometimes if you just focus the results on the patients that got the treatment as planned, that almost leads to an exaggerated result, and you don't want to be understanding and giving out treatments with exaggerated results. So we try and focus on the intention to treat population, which is every patient that was enrolled, not just the patients who actually got the treatment. And then there's statistics tests like log rank tests, which can help analyze all the data and all the forms that come in. So in terms of power, which endpoint is more important, progression-free survival or overall survival? Well, we've got to think, well, what does the patient want to know about? And what difference do we expect to show with our new treatment? And clinicians who are writing trials have to sort of hedge their bets before the trial begins, try and work out how much difference do we think this is going to make? Will it make a small difference, a big difference? And how many patients will we need to have the statistical power to actually show that difference? And there's lots of fancy calculations go on behind the scenes to work this out, but it all relies on knowing what is the endpoint that we're trying to show, and then what size of a difference do we hope to show. And if you get those things wrong at the start of the trial, you can end up with a negative trial, a trial that says this treatment isn't better, when in reality it actually was. But the way you designed the study wasn't efficiently going to answer that question from the outset. That's a clip from that song in the, was it 90s? I've got the power. Do you remember that? Anyway, no more singing. So the consort diagram is a great way to see every patient that entered the trial, what happened to them, how many patients got the treatment as planned, how many patients dropped out early. And this should feature very near the start of each paper to sort of show you and openly and honestly how many patients got the treatment as planned. And if patients didn't get the treatment as planned or they dropped out early, why was that? If lots of patients died in the first few weeks of getting the treatment, why was that? Was that just chance? Or was that because of some problem with the drug? And again, the consort diagram is really key to look at that. So that's a picture that you want to look at when you're reading a trial. This is a Kaplan-Meier plot, so basically a graph showing two populations of people within the study. The blue arm got durvalumab in this example after radiotherapy. The red arm didn't get any durvalumab immunotherapy after the radiotherapy. And you can see from the blue line being on top of the red line the whole way across, the patients who got the durvalumab had a higher probability of being alive the whole way across that graph. And at different sections there are different landmarks, we call them, sort of 1 year, 2 years, 3 years, 4 years, and 5 years. The numbers are there in terms of the percentage of patients who are still alive, and that helps you assess whether this drug is better than giving no drug. And in the numbers summary up in the top right corner is some statistical analysis to sort of convey how sure, how much confidence the authors had about this result. Because of course, this is only 500 patients. What if these results are just chance? What if it's just an unlucky day where more patients happen to die in one part of the trial than the other part of the trial? There's no way to know that. So statistics can be used to sort of work out what are the chances that this is a real result. And then forest plots, again, another fancy graph that you'll see in studies. And this sort of looks at risk factors for which patients might do better and which patients might do worse. And you can see that the blue dots, if they're on the left-hand side of that black line in the middle, if the blue dots are on the left-hand side, that indicates that that subgroup of patients benefits from the treatment. And if the blue dot is on the right-hand side of the black line, that would suggest that the drug didn't benefit that subgroup of patients. And you can see that across the board here, the blue dots are on the left of the black line, indicating that all subgroups of patients benefited from getting this drug. So again, you might say, well, sure, durvalumab, this treatment, was better than no durvalumab, but what about ALK rearranged patients? So I want to go in here and see where's the ALK line on this table. Actually, that wasn't included in the paper because that wasn't thought about at the start of the trial. But future trials and all the trials now that come out about lung cancer, they all have a line there for ALK rearrangement. So in the ALK population, is this result real as well? We know, sure, it exists, that real effect for all patients, but what about people with mutation in EGFR, people with ALK rearrangement? So you can see that now in studies. And that helps you as a patient, and it helps me as a clinician looking after a patient to know, is the patient in front of me today likely to benefit from this treatment? And then we have multivariable analysis. So there's another statistical test where basically we take in all the data we have and not just look at survival, progression. We factor in the patient's age, their comorbidities, other health problems that they've had, tumor factors, and try and work out, when you add all those things together, is there still a statistical benefit for that treatment? And again, that was very encouraging here. And you can see that ALK did make it in to this, this test in the Pacific paper. The discussion is the last part of the paper, and that's something I always try and read, because the authors have to come up with their interpretation of all the numbers that they presented in the context of the study design that they presented in the methods. They are bound to declare their weaknesses of the study. They'll have to say, listen, we didn't recruit as many as we hoped, or we didn't know about ALK mutations, so we didn't factor that into our analysis. And then they have to then plan what they'll do next in terms of advancing that research to make it even better again. So the discussion, if you don't understand the methods and the results, or there's stuff that you can't make proper sense of, going to the discussion is a nice text to go in there and read and try and understand what the results of the study are. And it should be written in a way that anybody can understand, and it will take into account the side effects as well as the main point of the study. The discussion tries to bring everything all together. So I think I put this slide in basically because if you're really stuck nowadays and there's some words you don't understand, I think, in my experience, I use it all the time. It's now on the front page of my apps on my phone. It's a great way for a 10-second explainer on terminology or a concept that you don't understand. And I think it's fair as patients and carers to use ChatGPT and other AI programs to try and break down information. Hopefully you won't rely on it too much, because we now have, thankfully, excellent resources, particularly for patients with specific mutations. And I know there's a superb new ALK-focused website that the Christie have launched or are about to launch. Hopefully I'm not giving the game away there, but I read about that earlier on, heard it earlier on. So, but I think ChatGPT and these other websites can be a useful way to learn about things if you're really stuck. I think it's also entirely reasonable to bring a question about a study or a treatment to your clinicians at your next appointment. Have it in your shopping list about things you want to go over. They might say, I'm not sure, I need to look it up myself. They might be going on to ChatGPT to answer the question, because clinical trials are a whole work of art, and the reason we have academic clinicians and academic oncologists is because research is really hard and it is really technical. And it needs to be, because it needs to be done right, so that we are approving the right treatments for our patients. And yeah, this is just what I got back from Pacific whenever I put that into ChatGPT. This was the basis of my talk. Someone laugh. I know about Pacific. So take-home messages. Nobody knows everything about clinical trials, including the doctors, so it's completely fine to ask questions. You also don't need to know everything about clinical trials. There's stuff in that paper I don't really fully grasp, and that's fine with me. I think if you do know something, if you read something that's useful, I think you should share it with someone who's in the same boat as you, because sometimes you do read things that are really useful, and that doesn't just apply to clinical trials and research like that, but less formal documents as well. And I think AI is a great tool. Trust but verify, as the Americans say. And I think if you're stuck, you should feel comfortable asking an expert, which may be your oncologist. Okay, so I've got 8 minutes left for questions. Thank you.
Randomisation. Have there been times when you've been in clinical trials and although you don't know what drug the patient's taking, you've seen one cohort working much better? Is there anyone, like the independent reviewers, who would know which group's doing which? So actually if there's an adverse effect that's not really working, that you can switch them over, or that's just part of the trial, it's the randomisation that's important?
Excellent question. So as clinicians enrolling to the trial, we're supposed to have equipoise, as in we don't really know which is better. So we're there rolling the dice with the patient. And some people like the idea of randomization. They think it's fair, and I'm just trusting this to the trial and the trial team. So other people don't like randomization. They want to be in the arm that gets that treatment, not that treatment. And again, that's something that it's hard to handle. In some trials, you know which treatment you're getting. In other trials, you don't know which treatment you're getting. You might not know, but your clinician knows, or you might not know what treatment you're getting and your clinician doesn't know what treatment you're getting. Your point about what if the clinician understands or comes to a realization about what treatment you're on, and that may be a good thing or a bad thing. I think sometimes the side effect profile of the treatment that you're receiving can give away which treatment you're in or which treatment you're getting. And I think that it's a subtle thing and you can never be sure, and it shouldn't influence the patient's ongoing participation in the study or the clinician's ongoing management of that patient in the study, in my opinion, because until all the results of the trial are completely concluded, you don't know, you can't really infer from this one person in front of you whether what they're going through is a good thing or a bad thing, if they should be in the trial or not be in the trial. And off the top of my head, one example, some of the immunotherapy trials for melanoma showed that patients got lots of quite difficult side effects from immunotherapy and lots of hospitalizations. And many of those trials, the patients wouldn't have known, or the doctors wouldn't have known what treatment they were in. The trials nurses would be none the wiser. But then after the trials were published, immunotherapy was extremely effective across the board. And one of the observations was that the patients who had higher rates of side effects actually got better benefit from the treatment. So that's just one example off the top of my head of how you really have to stand back and just trust the process, trust the protocol was right, and that that the truth will eventually come out effectively is the bottom line. But that's certainly a tricky scenario. I totally agree, yeah.
Hi. So two questions. One, which you probably just be able to say, yes, I share your frustration. As someone who sort of takes a casual interest in clinical trials, I find it hugely frustrating that there isn't a single worldwide registry. And I know clinicaltrials.gov covers an awful lot, but it doesn't cover everything. If a trial's not on in the US, it generally doesn't appear on that site. So it's very frustrating to try and build a list of trials that might be relevant to yourself as a patient. And I'd imagine it's equally frustrating as a clinician. Why is there not a single worldwide registry? Yeah.
I have not got a good answer for you, I'm afraid.
It's very frustrating.
I ask myself the question all the time. Clinicaltrials.gov is great for US. We've got UDRA in Europe,
yeah.
And there's others as well. And often the trials are registered on multiple or just one. It doesn't sit well with me either. I think just different continents like doing their own thing, and
you wonder what WHO's there for, if not.
Yeah. Yeah.
Yeah. So that was the first. The second question was really about, obviously one of the best times to get enrolled in a trial as a patient is at diagnosis, potentially, or progression, I suppose, but potentially at diagnosis. But it doesn't feel like it's part of the standard treatment pathways to think about it, and it's really down to the individual clinician. So is there more work that can be done in that space to make it part of the thought process or the standard treatment pathways that oncologists go through when they're taking on a new patient?
Yeah, I totally agree. You're preaching to the converted when you're speaking to me about this. I think there's an ambition nationally and beyond that every patient should have an opportunity of a clinical trial or a clinical study at least at every point in their journey. I think in an ideal world, that would be very achievable. The issues I think that mean that isn't actually the case is that it's difficult to open trials in terms of the paperwork involved and the resource for trials, because we work in a healthcare system where there is finite resource for these things. I think there's also an onus on the clinician to be aggressively hounding people who are slowing down the opening of trials. I think also because there's lots of different players in the research world. You know, we have academic trials started by clinicians, and we have pharmaceutical-driven trials, and you know, the agendas of both parties are very different often, and I think it's complicated in terms of which studies actually get to open. And so I would say that we need more investigator academic initiated trials, because often we're seeing the patients and we have these really interesting burning questions that we want to be answered. But whereas industry might be more interested in proving expensive drug X is better than less expensive drug Y, to be very cynical. But yeah, I think we haven't got enough resource for opening studies. There aren't enough good trials available. And then I think nationally also we have difficulties in terms of opening studies in the UK that have opened in other places in the world, be that Europe or the US or Asia. And I think all those things factor together. I mean, there's a lack of options for trials. I think unfortunately there's a really big geographical lottery at the moment. If you live in London, you know, there's probably lots more trial options than if you live in Belfast. So, you know, it's that factor's there. Not everyone can travel, and not everyone is able to travel or wants to travel. So it is a really difficult point. I think all you can do as patients and advocates is shout loud, write letters to MPs, and be angry about not having access to trials that your mate 200 miles up the road has access to. I think as clinicians, we do the same, basically. And I think the government needs to fund more academic positions for hospitals. Because if you don't have academic doctors working in the department, it's very hard to get trials open. I think the government's ambition to expedite the trial opening period, I think they have got a set deadline of April or so. From that point onwards, there's going to be a national dashboard where you can see how long it takes your trust to open studies, and the target by the government is 150 days. I don't know what trusts are like when you attend, but in Belfast it's many multitudes of 150 days to open a trial. So I'm very excited that if this initiative gets off the ground as planned and gets executed as planned, then that will make a huge difference in the next year, potentially. So that's one glimmer of hope.
Yeah, okay. I don't know if you can answer this question. Most of these trials, what I can see is they are all related to elaborate new target therapies. Are they trials in other type of therapy for ALK, like immunotherapy? So is there any research trying to see if ALK can be treated in a different way?
I believe there are lots of studies looking at how we should treat ALK or ALK mutated cancers. I'm not sure how many are open in the UK, but certainly when you go to conferences, World Lung was in Barcelona a couple of weeks ago. Deborah and I were there, and you know, there's all sorts of what seems like crazy stuff being presented by clinicians, stuff that's just completely different from what we do. And that's great, because until you do phase 1, phase 2 small studies, testing crazy ideas, if you don't do that, you're just going to always do the same treatment you always did. So we need folks to be designing experimental studies that are promising, that are biologically relevant, and we need patients to be brave and take the leap if they have the chance of participating. And I think it's only through small studies with really novel ideas do we find new treatment strategies and new treatment sequences. One example at the World Lung Conference was a whole session about, you know, what do we do with the primary lung cancer if a patient has metastatic lung cancer and maybe there's a mutation there in ALK or rearrangement in ALK or a mutation in EGFR. You know, should we be doing something with the primary tumor, even though there's other areas of disease? And classically, we've always thought that's, you know, there's no point. But now there's trials suggesting that there potentially is a benefit, but it's very, very early days. But what we need is a few good phase 1 studies, phase 2 studies, and that can help then build the preliminary data we need for a larger study to actually properly test the theory. And there's lots of other, what seem like very, very strange ideas being tested, but that's how new treatments get discovered. So, yeah, good question.
Yeah. Are placebos used in all phases of trials? Because that could feel quite risky for a patient.
I missed the very first word.
Can placebos be used in all phases of the trials? Because it could feel quite risky if you're going to be given a placebo, as opposed to maybe you've ended your TKIs, but you've now been off. You could either be gone to chemo, which could give you another 6 months, and then another treatment could be released, or would you try a trial and then be given a placebo?
That's a brilliant question. Phase 2 and phase 3 trials typically would use placebos to prove benefit if a treatment. If there's no other treatment left, they might say, let's try this and see if it's better than nothing. I think that strategy is sensible in some cancer settings. I think in the one you described, it's maybe not so sensible, but it depends on what the exact drug is for and what the setting is. I think that is one phenomenon about placebo-driven trials that does sometimes spook patients. I totally understand that myself on a personal level. But the trial would not have been approved or opened locally in your hospital if it wasn't deemed to be ethically right. So I think, again, there's all these questions and uncertainties, and you want to know, is it going to help you? You want to know, what are the side effects? But in trials, that's often not possible. And that, obviously lets anxieties creep in about what's going to happen to me. Is this going to be something that, doesn't help me? Maybe it'll harm me. I think you have to always factor in that this trial has been, looked at by hundreds of people from an ethical point of view, a scientific point of view. And in the UK, we are so highly regulated for clinical trials. If the clinical study was risky in that sense, it wouldn't be opened. That doesn't take away from
if you're not getting that drug, that's the risk. Because if you're getting the drug, then you've got a chance.
Yeah.
But if you're just getting a placebo, you're really not on treatment anymore.
I understand totally. I think, in the setting of metastatic cancer, there's, not so many placebo-controlled trials. But in other scenarios, it might be useful. For example, in the surgically operable ALK rearranged cancers, so people with localized cancer that hasn't spread, there was a trial looking at operating on that tumor, taking it out, taking the cancer away, and then randomizing patients to either getting an ALK inhibitor as a sort of preventative to sort of stop relapse versus no treatment, because there was a question about, is that drug useful in that scenario? Does it really actually help stopping relapse if you've had an operation for your cancer? That question was there. So the only way to know that, probably the most appropriate way to address that research question, is to give some people the drug and give some people the standard of care, which is no treatment after. So I think that trial was a placebo trial. The patient didn't know, the doctor didn't know whether they were getting the treatment or not. So where there is no treatment, a placebo-controlled trial maybe is fair. But I agree completely with you. If you've been on treatment for years, the idea of being on a placebo isn't too appealing.
I think that was the last question. So it just leaves me to thank Gerard for coming along. Thank you very much indeed. There's a small little token.
I didn't do that.
That's right. It's only little. So